Assalamualaikum warahmatullahi wabarakatuh. Here we are from group 2. We will explain about biological pesticide preparation. Next. Before that, we will introduce ourselves. In this group, there is ERIKA HUTASOIT number 064, and then me, LUHUNG BHAKTI number 188, and then MOHAMAD HAFIDH ADITYAWAN number 202, and DESTY ARTANTI number 233. Chapter 1 is Introduction, Background. Excessive use of chemical pesticide in the agriculture sector has a negative impact on the surrounding environment and humans. The natural balance will be disturbed by the emergence of resistant pests and parasites. One of the causes of the negative effect brought about by chemical pesticide is the presence of pesticide residues in the soil, which can poison non-target organisms, even being carried into stream to water sources, and can poison the surrounding environment. Therefore, an environmentally friendly substitute for pesticide is needed. And then chapter 2 is content. Definition of biopesticides. Biological pesticides are one way to control integrated pests or IPM. Biological pesticides are pesticides that are made and taken from natural ingredients. These pesticides are in great demand as a subtype for synthetic or chemical pesticide that can cause toxic side effects to both the environment and living things and can cause resistance to pests.
And then exploration of biopesticide for microorganisms. The first one, entomopathogenic fungi can be obtained by isolation from dead insect infected with the fungus from soil or from insect beating. The second one, antagonistic fungi can be obtained from fertile land that contains a lot of humus, land that contains plant residue, and from the soil around plants infected with soil-borne pathogens. The third one, nematodes and entomopathogenic bacteria can be obtained from the soil in the tillage layer around the plantation. And the last one, entomopathogenic virus are obtained from dead insect infected with the virus. Insect that die as a result of being infected with a virus that can be found on the surface of leaf, stem, or other organs in plants. The characteristics are black rotting dead larvae. Next, preparation of biopesticide. The first one, sampling. APH sampling in solid from according to SNI 19042 and APH sampling in liquid from according to SNI 190429. Sampling is carried by a competent sampling officer. Testing. The test is carried by a competent officer. Preparation of test sample in solid and liquid form. Three types of tests were carried out, namely the conidium density test, the conidium viability test, and the pathogenicity test. Packaging. Biological pesticides are packed in solid or flour, powder, and granule or liquid form. Packaging is made from materials that are not easily damaged and safe so that biological pesticides do not experience quality degradation.
Okay, so for next preparation of biopesticides is marking or labeling. So marking or labeling is written in a material that does not fade and is easy to read. Labeling at least includes information about first name and address of the producer. The next is types of biological pesticides or biological agents. The next is product form. The next is target pest. and conidium density, expired date, and then the last is production code. Next. Okay, so for next is biopesticides using viruses. NPV is a virus that is multifaceted and is present in inclusion bodies called polyhedra and replicates in the cell nucleus. The, the process of NPV infections begins with ingestion of polyhedra containing virus along with the feed in the digestive tract, which has an alkaline atmosphere. The polyhedra dissolves, releasing viruses or virions. Furthermore, the virus infects susceptible cells. Within 1 to 2 days after ingestion of polyhedra, infected caterpillars will experience morphological, physiological, and behavioral abnormal symptoms. Next. Biopesticides using bacteria. Bacteria can naturally suppress the growth of pathogens in soil. Several genera that have received much attention, namely Agrobacterium, Bacillus, and Pseudomonas. This strain and similar strains are referred to as plant growth promoting rhizobacteria, or PGPR. The term rhizobacteria in Pseudomonas (SPP), the ability of the PGPR as a biological control agent is due to its ability to compete for food substance or because of its metabolite products such as siderophore, hydrogen cyanide, antibiotics, or extracurricular enzymes that antagonize pathogens. Recently, it has been confirmed that Pseudomonas spp can stimulate the emergence of plant resistance to root pathogenic fungal infection, bacteria, and viruses.
Okay, next one is biopesticide using fungi. Entomology pathogenic fungi are one of the microorganisms that can be used as agents for controlling the main pests of soybeans. The most widely developed CAPS are Pneumorae, Riley, Behavior Bassiana, Lesanicillum lechani, Metarhizium anisopliae, Hirsutella thomsoni, and Paecilomyces fumosoroseus. The characteristic of dead as litura larvae infect with the fungus and rarely are colonies white mycelium 3 days after infections. The fungus will develop and colonize the whole body of the larva until it appears like green to the dark greens and forms a reproductive structure in the forms of conidia. And conclusions. Biological pesticides are pesticides that are made and take from natural ingredients. These pesticides are in the great demand as a substitute for synthetic or chemical pesticide that can cause toxic side effects to both the environmental and living things and can cause resistance to past biological pesticides, can also be viruses, bacteria, pathogenic nematodes, and fungi. Infective organ biopesticide derived from microorganisms in the forms of conidia or spores, fungi, polyhedral particles or viruses, juveniles or nematodes, and parasitors or bacteria. Steps to carry out the preparations of biological pesticide, namely sampling, testing, packaging, and labeling. Okay, that's all for our presentations. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.